Okay, I think it's time. Good morning and good afternoon. I hope you are doing fine and getting ready to start the fall after the summer break. So I'm Chieri Kubota. I'm a professor at the Ohio State University. I'm also the director of Ohio Controlled Environment Agriculture Center. I'm hosting this Indoor Ag Science Cafe, and then I like to have John Arlo with me. He is a PhD candidate at the Ohio State University in my lab, and then we do co-hosting this Indoor Science Cafe. So the Indoor Ag Science Cafe is funded by USDA NIFA SCRI program. And I know some of you are long-term contributors through participation of this cafe. But if you are new to this is a sort of communication platform, learning opportunities for conducting indoor vertical farm operations and also research in some cases, so that, helping each other type of platform. All right. So we didn't have any cafe July as a sort of standard practice. So this is sort of 2 months after the last one. Um, so the team is actually not just Ohio State, but Michigan State University, Purdue University, and University of Arizona. That's what we call OptimIA team, again funded by USDA. So let's start today's program. First of all, I'd like to show you the list of upcoming cafe series. We have already scheduled the cafes, and then this autumn or fall focus areas, I would say systems and sort of basics of engineering. So my talk today is not really engineering, or a little bit of engineering, but not so much engineering. But we have Ken Tran next month talking about lessons learned from the autonomous greenhouse challenge and how that experience and advantages apply to indoor farming. So that's indoor vertical farming. So that's quite interesting talk. October, we will have Robert Colangelo coming back and talking about a kind of interesting new system, modular, scalable farming system. So that's something we are looking forward to. He's founder of Green Sense Farms. And then November Cafe, we will have AJ Bose, greenhouse engineer at the Rutgers University, who is going to provide us basics of environmental sensors. You have sensors, but you probably don't know if they are using or you are using them in appropriate way to get accurate data. So that's something AJ is going to share. And then December, we will have another engineer faculty in UC Davis, Dr. Shamim Am, who is going to talk about, I believe it's a HVAC system and then sensor machine learning type of application, but it's going to be TBA, so we will know soon. So just look forward. Little bit more engineering type of topics than usual, but I think it's good to learn. All right, and then now one advertisement I have. OptimIA Group is building this educational resource we call OptimIA University. A lot of lectures online, free to access, and then all kinds of topics, including systems, little bit of engineering, plant response, cultivation practices, issues like environmental disorders and economics basics. So it's great materials. We are still working on some of the unfinished lectures, but those that are available are already available, already accessible. So please access and then learn something you didn't have that opportunity to learn. So that's I like to advertise. With that, I'd like to move on to the topic today. Unless John had something to add. No? Okay. All right, good. So John is going to moderate, and then this is recorded. And after that, we will have a lot of Q&A time, hopefully. So let's get started. So today's topic is why and why not harvesting lettuce crops early. And then I like to introduce an integrated evaluation we did for this topic. So I hope this is quite practical analysis and then relevant to your setting or situation. So again, my name is Chieri Kubota. I'm a professor at The Ohio State University. So OptimIA project is really to make indoor vertical farms more profitable, more productive, and sustainable. And then we have 3 objectives. So the 4 universities working together, right? Some of them are plant physiologists, some of them are engineers, some of them are economists. And then we are working together to achieve those outcomes under those 3 objectives. And then number 1 objective is defining and modeling the relationships among CapEx, OpEx, or capital expense, operational expense, and revenue generated based on yield and high value attributes for indoor leafy green production. Very important objective. And then for that objective, we have a great team of researchers and graduate students and 1 undergraduate student. We call modeling working team, and then or modeling working group team. And then this working group consists of 3 university members or 3 university PIs. Dr. Mura Kachira, who is a controlled environment engineer, specialized in modeling or sensor, remote sensing, and CFD. If you remember, he gave a really good lecture on CFD 101, computational fluid dynamics. And then his graduate student, Casey Chastain, who has expertise of computer science and crop modeling. So that team. And then the second team is Michigan State University Agricultural Economics team, led by Dr. Simone Valdesauza. And then her graduate student, Joseph Seong, at Michigan State University. And then my team, I usually attend solo, but I have undergraduate students who contributed. So I like to include her and then recognize her in this team. Lydia Krogi, and then she is, again, undergraduate student in our horticulture program. So we all together working multiple projects or topics. So we have a capacity to model not just crop growth, but economics analysis, modeling and simulation based. So we have a really great capacity to answer some sort of what if type of questions working together. All right. So this harvesting early topic, today's topic, is pretty much related with tip ban issue. In fact, I became aware of harvesting early as the standard practice in indoor vertical farms after we started working on tip ban mitigation practice or mitigation approach. But anyway, so let me start with tip ban. If you are not familiar with tip ban, so tip ban is the environmental nutrient disorder, and then it's happening when plants are growing fast. So that's problem because growers always want to have really good conditions that would accelerate plant growth. So that means yield is bigger under the optimum conditions. But whenever plants are growing too fast, this issue happens, and then usually very close to the end of the production. So going really well up to 5th or end of the 5th week or so of the 6th week, full cycle of the lettuce production. And then all of a sudden you see that tip ban. So growers want to avoid as much as possible because it shows up in a fast growing shoot tip area. And then basically marketability as a whole head is pretty much gone. Or you have to manually remove those dead tissues so that the rest of the plants you can market. It is calcium deficiency. Therefore, transpiration is a big factor. Therefore, so many factors involved to affect the tip pan incidence. So that's why it's a very complex problem. If you are not familiar with tip pan, actually OptimIA University has one lecture given by John Arrow talking about the basics of tip pan. So I would highly recommend watching that. And then something you may or may not know, I think tip pan can be more problematic in indoor vertical farm setting compared to greenhouse or even outdoor conditions. Why? Because typically inside vertical farms, indoor farms maintained with optimum growing conditions for the crops. And then also a very much limited airflow around the plants. It is not unusual to find airflow less than 0.1 meter per second. This is like 0.1 miles per hour. So it's very, very, very small amount of air moving around the plants. And it is not unusual to find relatively high relative humidity during the daytime, which is also causing more tip pan or increasing the risk of the tip pan. It is not unusual to see more than 70% relative humidity in the indoor vertical farm. And something you may not be aware, we call net radiation, so the total radiation of the sunlight spectrum, plus the infrared radiation or long wave radiation together. But we think under the sunlight conditions or the greenhouse situation, particularly if you target the same light intensity ppfd, micromoles per square meter per second over the crop, I think the net radiation is more in the indoor than the other way. Sorry, the net radiation is less in indoor vertical farm compared to greenhouse because sunlight has more energy, right? In addition to power, but electric lighting pretty much around power, so that the main light source doesn't generate too much energy over the plants. Therefore, it doesn't, electric lighting doesn't promote transpiration as much as sunlight does. So that's why, again, calcium deficiency, transpiration is very important. Less transpiration or lower rate of transpiration, that means more risk of tip pan. So there are multiple mitigation strategies. And very much practical one is just avoid, sensitive cultivars. However, this is a problematic approach because there are only few cultivars known as tolerant or resistant to tip pans. So it's not really good. So the second approach is practical, and that's exactly, I think, that most of the vertical farms are operating. It's just harvesting early because it shows up typically 5th or 6th week of the full head production. So instead of waiting to that stage, just harvest early so that completely avoid the risky timing of the tip pan. Another approach, which is more common in greenhouse, but also in a sense, indoor farms doing, just limit the potential growth by reducing light intensity or DLI, daily light integral, or lowering temperature. Again, this is not ideal either because it would reduce the productivity. And then the next one, enhancing mass flow, showing here, is a very much common approach in a greenhouse where you can have a big head space so that it is easy to manage airflow because downward airflow is necessary to enhance the transpiration around the shoot tip. To implement in the indoor vertical farm situation, it is not really, practical or easy to design that. So that's why I don't see this is implemented so much in the indoor vertical farms. Although we know that air circulation, airflow is very important. And then the last one is something we know it works at a certain degree, but growers do not like to continuously wet the plants with calcium chloride solution I think the frequency recommended is 3 times or 4 times a week, spraying with that solution. So foliar application may work, but it's not ideal approach. So yeah, so then indoor vertical farms typically harvesting early. So just recap, what is the strategy? So plants growing almost exponentially like that, right? And then if you wait until the fruit head forming stage, the per plant weight could be 100 grams, 120 grams. So that's a typical, the head lettuce, and then takes about 6 weeks or so. But the last 7 days to 10 days is very risky time because, as you can see, the slope of this exponential growth, it's very steep, right? So that means fast growing rate. And then in that rate, compared to the transpiration, the demand for calcium exceeding the supply by the transpiration, therefore the risk of tip pan increase. If you harvest early, right here, you can successfully avoid the tip pan risk. However, you have to increase the density. Otherwise, the per plant biomass or yield is such teeny tiny, probably less than 1/10. Therefore, you have to increase the density so that per area base, you can get a good amount of yield. In North America, most of the cases, growers are packing individual leaves rather than heads. So whether it is coming from relatively small heads or relatively large head, doesn't make a huge difference in terms of price point. I think that's another reason growers like to harvest early so that avoiding the tip pan, but they can still market as a packed, salad mix, out of their products. All right. So to justify that understanding, we did some mini survey. So some of you, if you responded the survey, thank you so much doing that. It helped us a lot to understand the actual practice and what's going on in indoor vertical farm and greenhouses. So one of the questions we asked was, how many days do you typically grow lettuce crop from seed to harvest? And then we had 13 responders. It's not that big numbers, but it was very helpful because it shows what it suggests clear difference between indoor vertical farms and in greenhouse farms, although the sample number is very, very small for greenhouse. But as you can see, from seed to harvest, it's up to 35 days indoor farms. But greenhouse beyond that, because greenhouse typically grow fruit head lettuce, and then indoor farms usually terminate at much smaller, so that 1 week or even 2 weeks earlier than usual fruit head stage, right? So that's what I'm talking about. So this working group, modeling working group, set this question: is a short cycle production more profitable than a full cycle production? So we took this integrated approach. So what we are integrating, number one, engineering approach, where University of Arizona, Mura Kachira and Casey Chastain combined image analysis and growth modeling to estimate planting density that produce the same annual equivalent yield when harvested in short cycle. So basically, what is the high density they have to achieve to be able to produce same amount of lettuce yield yearly base, right? Or whatever the cycle base. And then the second group, which is or second approach, which was conducted by my group, basically we quantified yield, biomass, nutritional quality, some of the nutritional quality, not all of them, but some of the critical nutritional quality, and post-harvest shelf life of the produce grown at high density short cycle production, and then compare that with the standard production cycle with a standard density. So that's what we did. And then the third approach is economic analysis using a simulation, using an economic model, cost model, Simone's group did the annualized revenue and operational cost analysis. So 3 together, we could conclude after this analysis. But anyway, that's what I'm going to show today. By the way, this topic was presented ASHS this year, American Society of Horticultural Science, presented by my undergraduate student, Lydia, beginning of August. So, all right. So the 2 slides I like to show out of University of Arizona approach. So they use camera, right, mounted on top of the or the above the plant canopy and monitor the canopy size, and then compute it top projected canopy area, like you can see. By the way, this face is the Casey's face. It's kind of interesting. I like this picture very much. But anyways, here is the camera, and then the plants are growing, and he analyzed that top projected canopy area, and then computed the biomass based on that information and existing lettuce crop growth model. The next slide is also from University of Arizona, showing that purple line is the top projected canopy area, and up to a certain point, it's almost following the exponential growth curve. And then as soon as the leaves are start touching, and then basically no more increase in terms of canopy area. So for this phase, you have to include the light interception, which is typical parameter for the growth model, so that it can predict the continuous yield increase during that phase. But canopy project area or projected canopy area, TPCA, can generate the radius or diameter of the plant. So that is very useful information for finding out the appropriate density at given time frame. So I asked the question, okay, what's the density we need to achieve to get the equivalent yield for 4-week cycle if I divide the 4 weeks into the 2 cycles of 14 days, so the 2 weeks and 2 weeks. So the short cycle, 2 times, and then equivalent to the standard 4-week cycle. And then they provided the answer, which is 214 plants per square meter density. And then the conventional reference point was 40 plants per square meter. But you see that result in a minute. So based on that information, we did the experiment at Ohio State. The same cultivar used as a material in one of the materials at University of Arizona, Surratt. And then we use our own what we call veggie box. That's a growth room with a shallow water culture hydroponic system. And then air temperature, light intensity, DLI, very similar. Light source was different from from what University of Arizona used, but that's what we had. And then photo period is the same. Humidity is a little bit higher in our condition during the daytime. So it's more tip-on inducing condition, I would say. And then CO2 concentration is about the same, but it's not exactly the same condition, but similar conditions we did the test. So mature crop cycle, usually, 2 weeks propagation time, and then 4 weeks of post-transplanting growth. And then that would give about 100 grams per head type of biomass, fresh weight. And then usually growers use 30s and 40s plants per square meter. And then we selected 40, so the 40 plants per square meter. And then short crop cycle at high density, we did the same propagation to the transplanting. And then the short cycle, only 2 weeks post-transplanting. Compared to 4-week cycle, because it's a short cycle of 2 weeks, we can squeeze 2 cycles in that 4-week time period. So later, I use 4-week equivalent yield to compare. And then that means the normal cycle, you have the harvest after 4 weeks. And then 4-week equivalent yield for short cycle, you have to add up. So the one cycle and second cycle total is going to be the 4-week equivalent yield of this type of production strategy. So you have 2 harvests, 2 transplantings, right? And then this is the density of the transplant production. All right, so the experimental conditions, again, 40 plants per square meter was the standard condition, 4 weeks. And then we set 3 different high density situations for 2 weeks short cycle production. Why 3 densities? Because we didn't know, one suggested by University of Arizona based on simulation is going to be the one actually, achieving the equivalent yield as the conventional 40 plants per square meter 4 weeks situation. Therefore, we set one density fewer than the one suggested by University of Arizona, and then another one, 272, greater than what suggested by University of Arizona. So this is the day, I believe, of the transplanting. So you can see the density difference. So the next slide is showing 1 week post-transplanting. You can see the size of the plants are pretty similar, right? 1 week after transplanting. And then some of the high density conditions, you see the beginning of touching leaves, so the closing canopy. And then 2 weeks post-transplanting, that's the time we harvested, Lydia harvested with some of other students' help, those high density short cycle crop, right? So 2 weeks, and then that's the end of the production of the short cycle. And then this 40 plants per square meter kept going 2 more weeks. And then I show you the picture. All right, so this is the 3rd week. This is the end of 4 week. So this is the time we harvested. And then it doesn't show clearly in this picture, but almost all the plants showed tip-pan, right? So our condition was relatively tip-pan inducing conditions. And then it just showed that successfully the concept, if you grow the fruit head lettuce in that kind of conditions, you get tip-pan. Therefore, harvesting early would successfully avoid tip-pan because none of them show any tip-pan. I have a data in a minute to show you. But so this is the size of the plant at the time of harvest. Again, those high density ones after 2 weeks. And then this is the after 4 weeks at conventional fruit head density. You can see that much bigger, 10 times bigger shoot mass compared to high density one. And then this is the value shown in the bar chart, bar graph at 40 plants per square meter density, you get about 100 grams. And then other high density conditions, it's about 1/10 of the yield per plant. Now, so this is per plant. So to get to the per production area, you have to multiply those numbers, right? Those numbers with density, how many plants per square meter. And then to get to the 4-week equivalent yield, you have to multiply this further by 2 because 2 cycles go into 4-week time period. Because it's a short cycle crop. So that's the result, 4-week equivalent fresh weight per production area per square meter. So the 4 weeks, so that's the 1 cycle, 40 per square meter density. And then you can see that compared to this reference point, some density like a 214 originally suggested by University of Arizona and 272 non-significantly different. So if you look at the 4-week cycle, or you can extrapolate a year-long cycle, you can get the same productivity, same yield coming out from the production area used after transplanting. So that's good, right? So let's look at the quality. Well, before going that, sorry, one more slide I wanted to show. Lydia did analysis, not just the upper part, but also root biomass. And then if you know plant physiology, you might have learned that, young plants tend to allocate more biomass to the roots. And that's exactly showing. This is at the time of harvest. So the after 2 weeks, high density plants, because they are young, only 2 weeks after planting, transplanting, therefore they tend to allocate more biomass to the root rather than shoot. So that is sort of waste because you're not going to eat roots, or you're not going to sell roots. So it's a small difference, but it's a significant difference compared to that 4-week growing cycle. So that's something you might want to consider. By the way, we have experiment 2 times, so analyzed uh, combined when there is no introduction to statistical analysis. And if there is any trend difference, then we show different individual data like this, experiment 1 and experiment 2. Tip pan, as I said earlier, tip pan was the case for the standard condition. And then the short cycle crop, because harvested early, which is a big advantage. That's why most of the growers want to do that. High density doesn't show any tip pan. We did a nitrate analysis. I believe Lydia found the literature showing that high density production tend to accumulate more nitrate in the leaves. So we wanted to see that if it is the case in this situation, but we didn't see that. It's about the same nitrate concentration. Nitrate sometimes a health concern. It's not regulated in U.S. government. But in Europe, sometimes it is problematic to sell leafy greens having a high concentration of nitrate. This is the post-harvest study we did also based on the literature suggestion. So we put the harvested leaves in a 4 degrees C cooler cooling unit, and then we looked at after 4 weeks, after 2 weeks and 4 weeks, in terms of the quality, visual quality, and then in this case, percent wilted leaves. And then as you can see, high density production, all of them having relatively higher wilting percentage compared to the full cycle 4 weeks lettuce, most likely because they are young, soft tissue. Because the density cloudness is relatively similar degree in our observation at the time of high density ending time, 2 weeks versus 4 weeks low density. So I think this is more like age related, but that's something you might want to consider in terms of post-harvest shelf life. And then I like to show the economic analysis. Dr. Valde Souza group did this simulation based. So what she did for us with her graduate student, Joseph, was taking that information, our information, yield, and then also conditions, light intensity and temperature, and then plug those information or those values into their models, and then compute it, how much area is required for growing transplants, and then also after planting, and then how much harvesting frequency happening in year-long basis, and then what is the resulting yield. So this is really powerful so that we can see the real impact of high density production. So let's take a look at that results of Michigan State University analysis. So this is showing annual cost and revenue per square meter total area. Total means the sum of propagation area plus production area. So earlier yield analysis we provided, that was a production area base. So after transplanting, right, the density is we tested was 40 plants per square meter to 272 plants per square meter density, as you can see in this table. That was the production area base. But Simone's group analyzed per total area base including propagation. Now, if you are doing high density production, right, that means you need a lot more transplants to grow. So the relative amount of transplant production area or propagation area is much bigger when you do high density production. So that's why this annual revenue is not even, well, not the same as the 40 plants per square meter condition. So the revenue is $751 per total area per square meter, but even at the highest density, it's only 660. So because more area is needed for the transplant production. And then operational cost. Well, before going that, so this is a sort of linear trend, right? So the increasing density, so this is suggesting if you increase the density further, maybe 300, close to 400, you probably get the annual revenue similar to that if the trend is maintained. However, the cost doesn't work that way. Cost increase that same way, right? So that means it's a negative direction. Why cost is much higher at high density production? So number one, I think it's a material cost, seed cost, substrate cost. You need more materials, and then also labor. If you are harvesting using workers, this can be automated, but if you're using labor to transplant or harvesting, that is basically doubling amount of labor cost. And then maybe some other ones, but the big factor was material cost and then labor. Therefore, the revenue, if I compute as annual revenue minus operational cost, that is a lot lower for the high density production, and then less than half of the conventional 40 plants per square meter, 4-week cycle. So this is showing that it's really challenging. Yes, you can avoid tip ban, but the revenue side it's not that great. The profit side it's not that great. So we really need to work on these conditions, to keep them out free from tip ban. So the environmental conditions, maybe air circulation, other approaches we are working on in a separate objective is so critical to make the indoor vertical farms more profitable and productive. So as a conclusion, so indoor vertical farm are likely highly inductive, inducive for tip ban, which limit the productivity and profitability for sure. And then short cycle high production or high density production, yes, you can avoid tip ban effectively. And then yes, you can achieve annual yield equivalent to standard production if you compare per production area base. And then also if you increase the density even further than we tested, likely you get a good yield, even including the propagation area. However, additional space requirement for transplant production and then also costs for intensive use of labor and materials, seeds and substrates, and then shorter shelf life may not be a good, characteristics of this strategy. So it is something we need to be aware of. Our integrated simulation approach, as you see this, very powerful simulation-based optimization approach. Now we can answer many what-if questions because we have growth model, we have cost and resource use model, and then we have a capacity of testing that theoretical suggestion using our facility in Ohio State. So I think it's a great capacity we established through this project. And then actually, one of very unique approach we presented 2 weeks ago at the International Horticulture Congress. Presentation was made by Casey Shasteen, the optimization of density at given cycle of production. So that 10 days, 13 days, 14 days, whatever. So you can find the best scenario, how many days to grow, what's the density to make the profit maximize. So it's very, very unique capacity. So I hope we will have opportunity to listen to what this the last topic I show in here showed recently. So anyway, thank you so much. All the members in my lab and then OptimIA members who specifically contributed to this topic. And then of course the USDA NIFA Specialty Crop Research Initiative grant. So I'm gonna stop. Thank you very much. I'm gonna stop recording here.